will discuss transcription. Please subscribe the channel so that you are able to study with me. Thank you. Transcription. So transcription is the formation of RNA from DNA. DNA has the information to produce many proteins in the cytoplasm. So DNA will transcribe the information to RNA, which will go outside the nucleus in eukaryotes and translate the message into proteins. Clear? So in eukaryotes, the RNA produced in the nucleus will go out in the cytoplasm and produce the protein. That message will be translated. Translation we'll study later. And in prokaryotes, since the nucleus is not bounded by the DNA is like this, it's a circular DNA, so it is not bounded by a nucleus, so it takes place in the cytoplasm only. Now what is a transcription unit? A transcription unit is having a area on the DNA. This is the DNA. So on DNA, schematic structure of this DNA is shown here. There is a promoter region. RNA polymerase is the enzyme for formation of RNA. So there is a promoter region. On the promoter region, RNA polymerase is going to bind, and the structural gene which will code for a particular protein, this portion of the gene on DNA in eukaryotes has so many different places where these structural genes are there. So these genes will code for some kind of protein, and at the end there will be a terminator which will be stopping the synthesis of RNA. This strand on which the RNA is going to be synthesized is called the template, and the other strand is strangely called the coding strand, because the complementarity rule, this RNA is going to match this. Okay, so if the sequence on this strand of DNA is say A, T, C, G, C, 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 so complementarity rule, according to complementarity rule, the RNA will have codes like this. So the complementarity rule here will be AUCG. Uracil will be the base instead of thymine. But the rest of the synthesis of RNA will be like DNA. But since the RNA is single stranded, the other strand will not be participating in the synthesis of RNA. Okay, so template strand is going to produce the RNA. Now the steps in the formation of RNA. First step, we conveniently divide it into different steps for our study purpose. Okay, so this is the DNA. Say at the portion which is identified, the RNA, the promoter region, the RNA polymerase, this is RNA polymerase, is going to bind, and the helix is going to unwind at this place. Unwinding of the helix. So initiation, RNA polymerase binds at the promoter region, and there will be unwinding of the helix. Okay, so what happens next? After unwinding of the helix, the RNA polymerase along with many factors going to start the transcription in the 5'3" direction. Okay, the next step is, many factors are going to help it in the transcription. Say this is the 3' end, so this will start in 5'3" direction only. Now what happens in the elongation step? The what happens in the DNA replication? The ATP, TTP, CTP, they are going to the raw material. Okay, they were the raw material. Same is the case here, but instead of thymine, AUCG will be the raw material here. So uracil will be there. Uridine mono triphosphate. So as per complementarity rule, they are going to come opposite here. So if this sequence say is A T C G C C G C, so complementarity will be U A G C triphosphate. Okay, same here. So they will sit here, opposite here. Okay, now pyrophosphate is going to get released, and the energy released will be used in binding of these, and these will be binded together. Okay, so on. So triphosphate will be converted into monophosphate, and there will be a chain formation. Now this chain formation is going to be here. This RNA polymerase will reach here at the end. After this, the unwinding takes place, means here the unwinding is here. When it reaches here, unwinding will come here. Okay, so at the end there is a termination site. At the termination point, this RNA polymerase will release the RNA like this. This RNA in 5'3" direction will be released, and RNA polymerase will be also released. There are many factors, termination factors, which will help in releasing of this thing. So this RNA chain is going to get released, and the helix is going to form again. So this is the structure of in eukaryotes. Transcription, the process is little complicated, and there is this RNA need to be processed. The process starts with splicing, and then there will be capping and tailing. Let's start. You know, the RNA produced in eukaryotes is called heteronuclear RNA because it has many sequences which are called introns. They will not be useful. Exons are the portions which are coding for the proteins. Exons will be used, whereas introns need to be removed. These are non-coding portions of the gene. So these heteronuclear RNA, hnRNA, will be cut or spliced to produce the again another RNA. These portion will be cut. They will like this with the first portion. This portion will be a form removed with the help of some factors, and this new RNA will be produced. Now this RNA again is not still messenger RNA. It needs to be processed. It needs a capping and tailing, means some factors at the 5' end will be added, a cap will be added, and at the end a tail will be added. Okay, of 200 300 adenylate bases will be added at the end, and initiation gap at the tip also, at the 5' also there will be some caps added, which help in synthesis or translation process, the identification between head and tail. So this is all about transcription in eukaryotes. Okay, now in the next topic we'll study genetic code and the types of RNA. Thank you. Please subscribe the channel and share it with the students. Now there are some slides to show you the whole process of transcription in details. Uh, the transcription unit is shown here, the promoter region, the terminator region. Then there is initiation process with along with you can see many factors. There is elongation process, the polymerase is at the end, and there is termination where DNA polymerase is released and the factors are also released. In transcription in process in prokaryotes and eukaryotes also, there is splicing and there is head capping and tailing. So this is all about these slides. Thank you.